Hello everyone, welcome back to my channel, and today we are going to discuss paper 1 variant 2 of May June 2025 from biology A level. This video is related to the MCQs part in which students usually make some silly mistakes, you can say. So let's see how we can figure out the answers by looking at the different options. So the very first MCQ is which row shows the possible uses of eyepiece reticule. So first one is comparing the diameter of two cells viewed with the multiplied 10 objective lens or calibrating a stage micrometer viewed with A into 10 objective lens. So what is a possible use? So I guess this objective lens, you must have done the microscope. So if we talk about the eyepiece from which we look into, so it could be used to comparing the diameter of two cells viewed with this one rather than the calibration. So the answer is B. Which statement about light microscope is correct? So light microscope uses light as a source to view objects. So what can be done? As the distance to see between two points as separate points decreases, the resolution also decreases. No, resolution has nothing to do between the 2 points. Number two, a bacterium 0.2 micrometer in diameter will not be visible if the resolution is 220 nanometer. Yes, it cannot be viewed. Two membranes that are less than 300 nanometer apart will be visible as the two membranes if the wavelength is this. No. This is not going to be the correct. The resolution will improve with visible light of longer wavelengths such as red light. No, it has no effect. So the next question says the electron micrograph shows a cell. What is the actual maximum length of a cell? So what you can do, you must have your paper with you. So I'm telling you the formula. I'm not telling you the correct answer. I'm just telling you the formula to calculate the actual length. So actual length is equal to the you can say size of image divided by the magnification. So whatever length you are getting from it, the magnification is also given. So you must have a scale with you. You will calculate the length in millimeter. Now what you need to do, you need to multiply it with 1000 to convert it into micrometer. Calculate the length and then divide it. You'll get your answer. Okay. So because I don't know what you are getting on your like millimeter scale. The table compares some biochemical molecule P, Q, R and S. So P has chlorophyll, DNA, RNA, but no peptidoglycan. Simple. Peptidoglycan is present in the cell wall of bacteria. So it means this one is not related to bacteria. Number two, chlorophyll is not present, DNA or RNA is present, and peptidoglycan is not present. So it cannot be bacteria, or and it cannot be like anything that can photosynthesize. R is it doesn't has chlorophyll, but it has these three. It can be bacteria because peptidoglycan is present in bacteria. And last one, it has only RNA. So let's see what it can be. So they have given us option like chloroplast, virus, mitochondrion, and bacterium. So the very first one, which has chlorophyll, which has RNA, and which doesn't have this peptidoglycan, so it cannot be viruses. Let's say it is not virus as well as it is not bacteria. So it can be among the two, the chloroplast or the mitochondria. So if chlorophyll is present, it would be chloroplast, sorry. So the option can be A or B. Number two, they have DNA and RNA. So in viruses, only the genetic makeup is present, so it can be virus and it can be mitochondria as well. Number three, that it has peptidoglycan, that is very sure for bacteria. So the correct option is this B according to what is given to us. The next question says So the next question says, which cell structure contain enzymes and are enclosed by a double membrane? So we are very well aware that what is double membrane. So very first thing that we usually read in cellular respiration, there is an inner membrane on which we have particles are present which help in the aerobic respiration. So definitely the answer would be mitochondria first of all. Second thing, chloroplasts have an outer membrane, and then there is inner membrane which is filled with some kind of stroma. Yes, it has the and nucleus has enzymes and enclosed by a double membrane. So all three are the one which fits these category. What is the correct order of cell structures? So first of all, we'll say it's organelle. Then organelle, we have cell. So what type of organelles are like can be arranged from largest to smallest? Let's say what is the smallest? So yes, ribosome can be the smallest and nucleoli can be the smallest. So we say that ribosomes which are present in the cell or on the endoplasmic reticulum are the smallest ones. So option can be B or C. The next says the lysosome on central. Central are quite bigger. They help in the cell division of animals. They're quite bigger, so lysosomes are smaller as compared to the centriole. Then nucleoli and then the centriole one. So the correct option is B. Next question is related to the different solutions, and there is a test. Which test do we use for reducing sugar? That you must know that is the Benedict test. Let's read what the question says. A sample of solution of sucrose tested for reducing sugar remains blue. It means that there is no positive test. But when another sample of same solution is tested for non-reducing sugar, the solution turns red, means the test is positive. It's a negative one. So the options are what explain the result. During the non-reducing sugar test, an acid hydrolyzes sucrose to glucose and fructose. Yes, this can be the reason. Number two, during the non-reducing sugar test, the sucrose molecule condense into polysaccharides. No, polysaccharides do not give the Benedict test. Then the reducing sugar test converts sucrose into no, the test does has nothing to do with this, but the acid actually hydrolyzes or breaks down it. The reducing sugar test hydrolyzes monosaccharide to disaccharide. No, so the only correct option is A. Which molecule contain at least two double bonds? Let's say we have collagen, we have hemoglobin, and we have triglyceride. So we have protein and we have fats. So what option can be correct? Which can have at least two double bonds? So saturated is not going to have, no, is not going to have the double bond, because saturated itself indicates that it is what it is, something related to single bonds. Okay, collagen do have and hemoglobin also have the saturated bonds. So here we have triglyceride. So triglyceride. So if we talk about saturated one, saturated has no bond, but triglyceride has. So the correct option is D. Next is amylose and amylopectin and glycogen are all polysaccharides. Let's say what are these polysaccharide made up of? Let's see. Enzyme X removes one maltose molecule at a time from the end of polysaccharide by hydrolysis of alpha 1,4 glycosidic bond, which shows how completely each of these molecule will be hydrolyzed by enzyme X, like one by one if they break down, what actually they break down into, what is going to be least hydrolyzed and what is going to be highly hydrolyzed. So first of all, let's talk about the most completely hydrolyzed. So amylose is the one which has that which can be hydrolyzed easily, while glycogen has a more branch structure, it cannot be hydrolyzed easily. So the correct option is D. Which property of water enable it to be a sufficient to be an efficient means of losing heat? Number one, the latent heat of vaporization of water is high. Okay, the heat capacity of water is high. Each water molecule can form a hydrogen bond with four water molecules, and the force of cohesion and adhesion on water molecules are very high. So C and D are cannot be the answer. The answer lies in the latent heat of vaporization or latent heat of capacity. Now here we are talking about the sweating. So sweating is caused by evaporation. So the only correct option is the first one. The latent heat of vaporization of water is very high. Specific heat capacity is related to how much heat it absorbed to raise the temperature by 1 degree Celsius. So which bond are found in the levels of structure of protein molecules? So what type of bond do primary have, secondary have, and the quaternary structure has? So the primary definitely has a peptide bond because the different amino acids are joined together by peptide bond. So when it roll into a secondary structure, it has the hydrogen bond, and further on when it rolls into the quaternary structure, there is a presence of ionic bond. So the correct option is this, C. Next question says prokaryotes and eukaryotes use extracellular enzymes. Extracellular digestive enzymes are found in the internal environment of some prokaryotes. Some prokaryotes are digested by hydrolytic enzymes during phagocytosis by eukaryotic cells. For example, if we talk about the amoebic dysentery, when amoeba enter the body, the phagocytes of our body engulf them and then destroy them, which shows the location where extracellular enzymes are produced. So site of production of digestive enzymes used by the prokaryotes, definitely they used inside them. Site of production of hydrolytic enzymes used by the eukaryotic phagocytes. So both are inside cells. They are not outside. They just gave you the concept of extracellular to confuse you. Otherwise, if you look at the statements that are given in the answer, the question is purely inside cells. Next question says the amylase break down starch molecule. So we have already done that. There is an enzyme that first, which is easily composed or which are difficult to compose, that is related to amylose. Which substances will have the same number of molecule for the duration of this reaction? Amylase, water, maltose, and amylose. So what would remain same? So the enzyme or the concentration of enzyme remains same during the reaction. They are not going to use up, they are only there to speed up the reaction. So that enzyme that is going to exist is this amylase enzyme. Which graph could shows the effect of pH? So pH is moving from 1 till 14. So we say first of all the reaction increase, and then if the pH is not suitable, it can get denatured. So this too is very correct. Denature mean the activity will decrease. So definitely these are not going to be the part. So the answer is 2 and 4. So this is all for this video. Let's discuss the other MCQs in another video. You need to watch that video for your answers. If you need any explanation, detailed explanation, or any of the question or from any of the paper, you can ask in the comments. Thank you so much. See you in the next video.